time-resolved results for nanoparticle analysis.
Nanoparticles, lasers, and liquids. How do these go together? Well, like you, I'm full of questions. Hello, my name is Keith, and today I want us to have a look into the research of Professor Tobias Madl from the Gottfried Schatz Research Center for Cell Signaling, Metabolism and Aging, Molecular Biology, and Biochemistry at the Medical University of Graz. So Tobias, it's great meeting you. You are researching the dysregulation and drug targeting of biomolecular condensate formation. Can you please describe to me briefly what your studies are used for?
So the systems that we are interested in are biomolecular condensates, build up of proteins and protein RNA most of the time. And these complexes they form and have important implications in cell physiology.
So what are some of the biggest challenges when undertaking your research?
So actually one of the biggest challenges to study the formation of these condensates with molecular and with size resolution, and also the change of these condensates over time, how ligands, how drugs modulate these condensates on site and in real time.
So which methods did you try before changing to OF2I to address this particular challenge?
Yeah, in my lab we use mostly NMR spectroscopy, other biophysical techniques like small angle X-ray scattering. We use a lot of fluorescent-based techniques, microscopy and so on. They all have their pros and cons, but also severe limitations.
So why have you been dissatisfied with some of these methods?
Yeah, there is a really problem with starting the early phases of condensate formation. So when these condensates are very small, you have proteins interacting with RNA, they start forming some small complexes. These complexes get bigger and bigger and bigger. In particular, these early stages are interesting for drug discovery. And we are more or less blind in this early phase, so microscopy is not possible in resolving these early condensate formation processes. So that's not the only problem, so the size is a problem. The other problem is that we need high protein and RNA concentrations for the studies. Often material is limited, in particular if you work with patient samples. Another important aspect that inside the cell, the concentration of these proteins is also very low. So we would like to study actually these processes at physiological protein concentrations. And on top of this, it's also very difficult to study all of these processes over a wide size distribution in a time-resolved manner.
I understand. So what happened next, and how did you get to know BRAVE Analytics?
Yeah, that was actually a funny story. So we are working together in the same research center, and it happened that I visited a seminar of Christian Hill that he gave in the framework of our regular seminars. And he was talking about this very exciting technology to study particles in solution with size resolution. And we were talking to each other after the seminar, and we found out that actually it might also be possible to study these very exciting condensates that we're working on. So we took a sample, stopped by at Christian and his team, did some measurements, and were really struck to see that we can study these condensates with size resolution in the blind area that we have never actually resolved so far with time resolution. So it was really cool. We did a lot of experiments with proteins involved in neurodegenerative diseases, in viral infection, in SARS-CoV-2 and herpes virus infection, in diabetes, so to look at the aggregation condensate formation processes of all these proteins. So that was actually the starting point now where our science will follow up along these lines. Basis is set for very exciting follow-up studies also in context of drug discovery. And of course, we also look forward purchasing the instrument and expanding our research along these lines.
Wow, I mean, sounds like a perfect match. Thanks so much, Tobias, for this small excursion into the dynamics and importance of biomolecular condensates. It would be amazing to get some deeper insights for handling senile dementia, Alzheimer's, and cancer, if I can ever remember it. But let's see how they solve this particular problem.
This is Doris, Laboratory and Application Specialist at BRAVE Analytics, and she'll show you how to operate the BRAVE B-Curious Laboratory device. For every measurement, basically these steps must be taken. Find out the appropriate dilution factors for your sample. OF2I is a precise counting method and characterizes samples from ultra-low concentrations, like in water analysis, to solutions with about 10 to the power of 8 particles per milliliter. Thanks to the active and flow-through measuring principle, samples in this range can be measured directly. No further sample preparation, such as separation or concentration steps, are necessary. For higher concentrated samples like emulsion products, HANS, the BRAVE software, helps you to find the perfect range for your measurement since particle concentration is continuously determined. Then, you load your sample into the BRAVE B-Curious laboratory device. Clean the measuring tube and introduce it into your sample. Choose your preset measurement method and enter sample description. The intuitive HANS software will guide you through. If necessary, trigger the dynamic reaction of your sample and press start. Now the BRAVE B-Curious works its magic. You can observe your particles and results immediately. In the main window, HANS instantly calculates size and number-based size distribution. He shows you the detected single particles as stripes. In the bottom graph, you can see the particle concentration and d-value statistics over time. The measurements can run continuously from minutes up to hours to track your processes.
Amazing, isn't it? Particle size distribution and the dynamics of nanoparticles in liquids are not the only results which the active measurement principle of OF2I can deliver. You get a feel for complete processes within biophysical systems. Wow! Knowing the BRAVE team, I'm sure that's not all you can do with this small but effective lab instrument. I look forward to exploring more application examples together with you guys.